So the question is that in an iron overload state like hereditary hemochromatosis, hepcidin level is low. And hepcidin level is also low seen in iron deficiency anemia, which is the iron deficiency state. So what's the explanation for this paradox? And in this session, I'm going to particularly focus also on what are the changes in the lab diagnosis of the hereditary hemochromatosis, how the changes have been made nowadays with the advent of the newer sophisticated test. So firstly, I would start with the concept of hepcidin. Hepcidin, you all know that it's a 25 amino acid containing peptide. It's a hormone which is synthesized by hep. The name gives you a clue. It is synthesized from the liver. And previously it was thought that it has only bactericidal property. That is the main reason. So that's why they gave it hepcidin. But later it was found that its more important role is basically in controlling the iron homeostasis, controlling the iron metabolism inside the human body, particularly the iron absorption. So coming back to the basic fact, hereditary hemochromatosis versus normal, what is the difference? Normally what happens that if your body's iron level rises, hepcidin level would also rise. And when hepcidin level rises, it would help in stopping iron absorption from the duodenal enterocyte. How? To explain this, let's think that this marker is the duodenal enterocyte. This side is the lumen and this side is the basolateral side, is towards the body, towards the inside of the body, towards the blood vessel. Now, what is happening towards the lumen? You know these basic things. I think maybe 6th or 8th standard you have read. The iron metabolism is tightly regulated. Iron absorption is tightly regulated because iron cannot be lost from the human body. We don't have much avenue to lose iron from the human body. Only we can lose it through sweat. We can lose it through exfoliation of the epithelial cells. We can lose it through menstruation in a female with reproductive age. So we have a limited avenue to lose iron. But so body controls iron metabolism through iron absorption, and those molecules which you have read basically at the basic level, maybe that these are the things which are there, that in the luminal side there are two molecules. One is called duodenal cytochrome B, which converts the Fe3 to Fe2 plus, because Fe2 plus gets inside the duodenal enterocyte through the bivalent metal transporter, or 1, because Fe2 plus only 2 gets into the duodenal enterocyte, and once it gets inside the enterocyte, it can be stored inside the ferrotin. But I would now focus on the more important molecule, which is on the basolateral surface. There are a lot of molecules, hepcidin and other molecules are there, but more important molecule is the ferroportin, which is there towards the basolateral surface. This ferroportin molecule basically channelizes. It's a ferroportin, iron ferroportin. Port means gateway, entry point, door. So basically it channelizes the iron which is absorbed through the duodenal cell into the human body. Now this ferroportin molecule has a interesting relationship with the hepcidin. Basically the relationship is inversely proportional. They don't like each other, they hate each other. If one goes high, another goes down. You understand? That kind of relationship is there, like North Korea, South Korea. That means normally what happens when the hepcidin levels are high, ferroportin level is low. And when the hepcidin levels is low, ferroportin high. So hepcidin basically controls the expression of the ferroportin, particularly in the duodenal enterocyte and also in other cells like hepatocyte and macrophages. Now what happens normally when a person has enough or sufficient amount of iron in the human body, hepcidin level increases. So when the hepcidin level increases, what would happen? These ferroportin in the duodenal enterocyte, their level decreases. So now not much iron can be channelized inside the blood vessels, through the blood vessels into the human body. They cannot be channelized inside the human body, you understand? They can be absorbed in the duodenal enterocyte, but they would be not channelized into the blood, and they would not be bound with transparent other things, because the ferroportin, their level is decreased due to the high level of the hepcidin. This is happening in a normal state when you have high or adequate amount of iron. But what is different in hereditary hemochromatosis? The name tells you that it's a hereditary state. So obviously it's a genetic disorder, typically autosomal recessive disorder, and this there are a lot of subtypes are there, type 1, type 2, type 3 there. Most common one which we get is type 1, which is typically due to a mutation in the HFE gene. HFE gene located on the chromosome 6. And there could be mutation of other genes also in the other subtypes like homojuvelin, HFE gene, it's the hepcidin gene itself could be mutated. The outcome of this mutation, particularly the HFE gene, is that hepcidin level remains persistently low in the hereditary hemochromatosis patient. So if their level remains persistently low in the hereditary hemochromatosis patient, what is going to happen? Even if your body's iron level is adequate or high, what is going to happen? Hepcidin level is not going to rise, because as a result of those mutation, hepcidin level is always low. Now what is going to happen to that molecule I was talking about, which is there on the basolateral surface of the duodenal enterocyte? That molecule is ferroprotein. So ferroprotein, I told you, they have a inversely proportional relationship with them. So when their level is low, hepcidin level is always persistently low, ferroprotein's upregulation would occur, their level would be always high, they would be highly expressed on the duodenal enterocyte basolateral surface. So they would be causing always increased absorption of the iron inside the human body. They will be channelizing the iron through the duodenal enterocyte inside the human body. And this is happening in an uninhibited manner, in an uncontrolled manner, without any controlling mechanism, without any feedback mechanism, because hepcidin is always low, persistently low due to the genetic mutation is occurring. So as a result, what is happening in hereditary hemochromatosis, that due to those mutation, particularly HFE gene mutation, C282Y or H63D, these are the two most common mutations which you see in the HFE gene, or also less commonly could be other genes also. As a result, the hepcidin level is low, persistently low, always low. Always there is a high level of ferroprotein, and they are controlling, causing increased absorption of the iron in the human body. They're channelizing it inside the human body. That is the main problem. So the main problem in hereditary hemochromatosis is systemic. It's a systemic disorder which is characterized by uncontrolled overabsorption of the iron due to persistently low hepcidin level. Now, hepcidin level low, you'll be getting in another condition also, which is totally opposite of that, iron deficiency anemia. But iron deficiency anemia patient is not having any genetic disorder like this, hereditary hemochromatosis. What is happening there, as your iron deficiency state is there, body thinks that we need to right now absorb more amount of iron, so we need more expression of the ferroprotein in the intestinal duodenal enterocyte. Now how it would happen? By making the hepcidin level low, because if the hepcidin level is low, then only the enterocyte, the duodenal enterocyte, could overexpress or upregulate the expression of the ferroprotein, so that more and more amount of iron could be absorbed. So this explains the paradox here actually, that iron deficiency anemia you also get low hepcidin, and hereditary hemochromatosis you also get low hepcidin, but due to different reason and different outcome actually totally. Now the second thing which I want to discuss here, that hereditary hemochromatosis, there is not much role of liver biopsy or pulse stain, all these things are there. This role has been limited. Nowadays the diagnosis of hereditary hemochromatosis is basically based on three things. One is the presentation, family history, all this kind of thing, because it's as I mentioned, autosomal recessive disorder. Second thing, we usually do iron studies, and iron studies, what we typically get, the ferritin level is very high in hereditary hemochromatosis, and transferrin saturation would be very, very high also in the hereditary hemochromatosis. So these two things, if it is very high, normally transferrin saturation is 33%, in this patient we get more than 45%. If these two things are high, we straight away go for genetic analysis. And as I mentioned, there is a characteristic gene, HFE gene mutation, particularly C282Y or H63D. These are the two typical mutations we get. So if these things are done, there is not much role of previously, as we used to do the quantitative measurement of the iron in the liver biopsy, or doing the liver hemosiderin stain, looking for pulse precision blue. These things actually have not much significance right now. It's basically presentation based on that history, presentation, iron studies, and genetic analysis. This is the way these two things are done. And this also explains that why the main problem in hereditary hemochromatosis is low hepcidin. That is the key problem due to genetic mutation, and that's causing the increased intestinal absorption. So I'll continue this in the further sessions also at different time. Thank you so much.